Hi, this talk is about the brain stem. So the part of the brain stem first. Here you have a sagittal section of a dog's head, where you see the encephalon right here with the cerebrum, the cerebellum, the brain stem, and continues as the spinal cord. So now we will focus in these two images, and I have marked in colors the different parts of the brain stem. First, in red is the diencephalon. Then we have in green the mesencephalon, in yellow the pons, and in blue the medulla oblongata. There you have a dorsal view of the brain stem. What are the functions of the brain stem? There are many. They control the cranial nerves. It serves as a pathway for ascending and descending vias from the spinal cord to upper centers and from upper centers to the spinal cord. It fights against gravity. It initiates movements that are not particularly skilled and regulates vital functions and activates the cerebrum through the ascending reticular activating system. Let's have a look at this video. In this video, I have put an encephalon inside the cranial cavity, and we are going to make transverse sections. In the first, we do not see the brainstem yet, but we identify structures that we know as the coated nuclei, telencephalic septum, corpus callosum, white matter. This is the fibers, projection fibers, the internal capsule. And also we identify down here the optic nerves, the cingulate gyrus, one and the other. We see fibers that are going to become the rostral commissure. Here we still see the optic nerves very close to the optic chiasm. Here the sella turcica with the dorsum sellae that holds the hypophysis. Look how the dorsum sellae is introducing between the crus cerebri of the mesencephalon. Here we have the pons and the medulla oblongata. Okay, easy to identify structures. Okay, second section. This section has been done at the level of the hypophysis, you see, forms part of the hypothalamus. We see both thalami joined by the interthalamic adhesion here. This is part of the fornix, hippocampal formation there and here. Next section. This section was done at the level of the dorsum sellae. So we see the mesencephalon here, okay, with the hippocampus. The other, as you see, there is limbic cortex, part of the thalamus, of the geniculate bodies of the thalamus, the mesencephalic aqueduct, the rostral colliculi. This is the corpus callosum, and here part of the fibers that get out from the hippocampus. And particularly, we are very close to the splenium of the corpus callosum, the crus cerebri, that form the ventral portions of the cerebral peduncles. Look, this is interesting, this video, because it shows you that the brain stem is located occupying the middle fossa, the caudal fossa, okay? But there is a lot of space in front of the brain stem here. In the next section, we have cut at the level of the caudal colliculi of the mesencephalon. We see the vermis of the cerebellum, and we see pons here. As you see, this is located in the caudal fossa of the cranial cavity. Now we have removed the cerebral hemispheres, and in the next section, we are cutting medulla oblongata, right here. We will study these structures later on. Part of the vermis, cerebellar hemispheres. Look, that most the brainstem is located quite caudally. Well, we have here the diencephalon, but caudal to the sella turcica, we have mesencephalon, pons, medulla oblongata. The pyramids. Now, for the next section, we are cutting medulla oblongata. You see, this is the accessory nerve. We will study this nerve later on in another talk. We are at the level of the orbix here. Okay. So now you have the idea where the brainstem is located in the cranial cavity. Let's study the parts. The first one is the diencephalon. And the diencephalon is formed by the epithalamus, thalamus, hypothalamus, subthalamus, and mesothalamus. Look, that all this there is one structure that is it is not in the middle, but somehow there, and all the rest of the parts of the diencephalon take their name according to their position to the thalamus. Thalamus meaning internal chamber. Okay, so thalamus. Remember that there are two thalamus. Okay, one right and one left, and they are united by the interthalamic adhesion. So it's a pair structure. However, the hypothalamus is not pair, but the thalamus is a pair structure. Okay, let's start with the epithalamus. Oh well, the thalamus, better. And we will, from there, we will go to other parts. The thalamus. The thalamus, both thalami are quite big structures and full of elevations that correspond to nuclei. Here, this is a dissection of a brainstem. I have removed the cerebral hemispheres. So here you have the optic nerves with the optic chiasm and the optic tract that runs up to the lateral geniculate body of the thalamus. And medially and more ventrally located is the medial geniculate body. On a rostral view, we identify other structures, the columns of the fornix there, the rostral commissure, right? And the columns of the fornix running caudal to the rostral commissure. This elevation here corresponds to the rostral nucleus. On a dorsal vision, we see the left thalamus and the right thalamus, right? And this is the third ventricle. The rostral nucleus is this nucleus here, the lateral dorsal nucleus, the lateral caudal nucleus, the lateral geniculate body. And more medially, you have on the caudal position, the pulvinar, that means from Latin pillow, the dorsomedial nucleus, and the paraventricular nucleus. Okay, if we open carefully both thalami, we identify the interthalamic adhesion here. Here we are at the level of the third ventricle. Now, let's try to identify these nuclei through sections, through transverse sections. At this level here that is marked on this image, on the superior left image, we have cut and we identify the interthalamic adhesion. This is hypothalamus with the mammillary bodies right here. Here in white, we have part of the columns of the fornix and the mammillothalamic tract here. And we identify there is a collection of cells, of somas, of bodies of cells here. That is the paraventricular nucleus. We also identify dorsally here the dorsal medial nucleus, the lateral dorsal nucleus. Here we have the ventral rostral nucleus. This nucleus cannot be identified from outside because it's hidden. And more laterally located, it is the reticular nucleus. All these nuclei receive different information from different parts of the central nervous system. If we make a transverse section a bit more caudal, we identify the lateral dorsal nucleus. Dorsomedial, we see the paraventricular nucleus. Again, the interthalamic adhesion and the lateral ventral nucleus. And the structures we have identified previously, and also the reticular nucleus. We are more caudal now. We are at the level of the methothalamus. The methothalamus is the part of the thalamus that is formed by the lateral geniculate bodies and the medial geniculate bodies. Okay, we have the lateral geniculate body is this structure here, and it seems to be divided into portions because all these fibers, white fibers, belong to the optic radiation. Okay, the fibers that leave the lateral geniculate body in order to form part of the internal capsule, and they reach the occipital cortex. Here we have the medial geniculate body. This is the lateral, and then we have more ventrally the medial geniculate body. And this white matter here is the acoustic radiation, fibers that from this nucleus, they reach the temporal cortex. And again, this we have the ventral caudal nucleus. These are other views of the thalamus. And as you see, the lateral geniculate body, it's connected with the rostral colliculus of the mesencephalon through the brachium of the rostral colliculus. As well, the medial geniculate body is connected with the caudal colliculus of the mesencephalon through the brachium of the caudal colliculus. Look at the arrows. It means this arrow means that this arrow goes from the lateral geniculate body to the rostral colliculus, because the information first has reached the lateral geniculate body through the optic tract. And some fibers go to the cortex, but other fibers for reflexes, for turning the head towards a visual stimulus, these fibers reach the rostral colliculus of the mesencephalon. From there, the tectospinal tract will send information to cervical spinal segments in order to turn the head towards this visual stimulus. So this is the direction of the fibers. However, here we have pointed the arrow rostrally. It means that the caudal colliculus is involved in auditory reflexes. So fibers from the caudal colliculus are going to go down, forming part of the tectospinal tract in order to reach cervical spinal segments for turning of the head towards an auditory stimulus. So in this case, these fibers that have to be conscious, they have to proceed rostrally and reach the medial geniculate body through the brachium of the caudal colliculus in order to be projected to the temporal cortex following the acoustic radiation. So again, this is a fixed encephalon of a dog. And then you see here, this structure is this one here, is the lateral geniculate body. And as you see, fibers of the optic radiation. And then the medial geniculate body with fibers of the acoustic radiation. So when we identify lesions in the brainstem affecting the thalamus, if we know which nuclei could be affected, we may have clues. However, it is not easy, right? So in this case, the lesion may affect the reticular nucleus and the ventral lateral nucleus. So this lesion here, this lacunar lesion here, it affects what? It affects this area. This area is comprised a bit more ventromedial to the lateral geniculate nucleus, so maybe affecting the ventral caudal nucleus. So this is another as a bigger lesion, right? And in this case, it is affecting, well, a lot of nuclei from the dorsomedial nucleus, ventrolateral, and paraventricular nucleus. In this case here on the right, you are cutting the brainstem at the level of the mesothalamus. So we are at the lateral and medial geniculate bodies. So, and also a bit more ventral. So this portion of the ventral caudal nucleus may be damaged, and also part of the lateral geniculate nucleus. So what's a thalamus? A thalamus, it could be as a big train station that all the trains that are taking information from different parts of the body, they have to stop here in order to reach the sensory cortex. So what is going to happen when there is a lesion of the thalamus? So we may be wrong. It's like waiting in here at this end station, and we see that the trains that should be coming this way, they are not coming. So we first think that something must be wrong at the origin of the line. But no, the problem is at the thalamus, at this train station like, right? So it's a way of looking at the thalamus, a very easy one. So now we go to the epithalamus that is on top of the thalamus. What the structures are, sorry about that. What the structures are on top of the thalamus? We have the pineal gland, okay? The abenula. What is the abenula? Abenula means restraints. And this is a structure, okay? Here we have the dorsal view of the brainstem. This is a transverse section at this level. So here, and then this square has been enlarged. In order to identify, look, this is the abenula. What do we have caudally here? We have the caudal commissure that has been sectioned. Remember, the caudal commissure is related with the fibers of the optic tract. Under the caudal commissure, we have the subcommissural organ. This is a circumventricular organ, secretory circumventricular organ. And the abenula. And the abenula is connected with, here, you see the abenula in this section, here, you see the abenula. And this fasciculus, there's the abenulo-intercrural tract, also known as fasciculus retroflexus of Maynard. This fasciculus connects the abenula with the intercrural nucleus of the mesencephalon. And also, the abenula is connected through the stria abenularis thalami. This way here, this pathway, connects the abenula rossuli, located here, dorsal to the third ventricle. This is the third ventricle, or the diencephalic ventricle, and connects it with the telencephalic septum. Here you have a summary of all the pathways that form the limbic system, okay? So we have seen that the abenula is connected through the stria abenularis with the septum and the accumbens nucleus. This is a basal nucleus, okay? Remember, as well as the hippocampus is connected through the fornix with the septum, the accumbens, and the mammillary body. And also we have the stria terminalis that connects the amygdala. We have seen the amygdala that was enclosed where? Inside the piriform lobe, remember, from another lecture. It connects the amygdala with the septum, telencephalic septum, the accumbens, and the mammillary body. However, the stria terminalis is connected a bit laterally to the thalamus, at this way here. Okay, this is thalamus, and this is abenula, so it's lateral to the thalamus, the stria terminalis. Okay, so we get to the pineal gland, that means like a pine tree, or epiphysis cerebri. Remember that René Descartes thought that the pineal gland was a valve or a connection between our soul that was in the brain and the surrounding media. There was a kind of connection, right? And has been studied in esoteric sciences. However, if we look in anatomy, this is the pineal gland, like a pine, of a dog. You see, this is the third ventricle, one thalamus, the other thalamus, the rostral colliculus of the mesencephalon, and the caudal colliculus, and the other one, caudal colliculus of the mesencephalon. And here we have the pineal gland. In the case of a bovine, it's much bigger. What is the pineal gland? The pineal gland is a gland that produces, is very vascularized, right? And also is very well innervated by sympathetic nerve fibers. So the pineal gland produces a lot of hormones. However, one of them is the serotonin that is going to be transformed in melatonin. And in this case, the light inhibits the production or the transformation of serotonin and melatonin. So in the conditions of darkness, we have more melatonin, so we have sleepiness, and is an antigonadotrophic activity and restrains sexual development. Okay, so so this pineal gland has to be connected with the light. Here you have the pineal gland of a sheep. Look at it. It's very, very big. We have the rostral colliculi, the left, the right, the cerebellum, right here. We have opened the corpus callosum, right here, the fornix, in order to identify the most dorsal portions of the brainstem. And you see the pineal gland, and these connections is the stria abenularis. Here. Look how big is the pineal gland in a sheep. And also you identify other structures. This is the hippocampus here, part of the fornix, hippocampus again, right? It's a limbic because it's in the border of the brainstem, as you see, in the border of the brainstem. So, as in birds, it seems that the light and small vertebrates, the light goes directly through the roof of the cranial cavity to the pineal gland. However, in mammals, This light comes from the eye, runs with the optic nerve up to the optic chiasm, optic tract, and then reaches the suprachiasmatic nucleus of the hypothalamus. From there, it is not very well established how it reaches cervical spinal segments T1 up to T4 to the intermediate gray substance that are sympathetic. So preganglionic neurons reach the cranial cervical ganglion, and from there they enter the cranial cavity and they reach the innervate, the pineal gland. Let's go to the subthalamus. Okay, the subthalamus is formed by two portions, the subthalamic body and the zona incerta. Okay, where is the subthalamus located? Look, this is a transverse section of a brain stalk. You see both thalami, the interthalamic adhesion, hypothalamus here. So part of the hypothalamus is located here with the crus cerebri, the optic tract located there. You remember the hippocampal formation, okay? And then you see, well, this is the avenula here, the fascicular retroflexus, you know many things now. So now this structure here is the subthalamic body and the zona incerta that extends dorsally to the crus cerebri. Do not be wrong with these portions. These are tracts, right? Remember that we are in a Balchowski, so we see the more brownish is fibers, okay? The mammato-tegmental tract and the cerebello-rubro-thalamic tract located here. Okay, and the subthalamus, as we have seen previously in other lessons, it's very important for the movements, to control movements and posture. And we will see that in another lessons, how important is the subthalamus. Let's go with the hypothalamus. Hypothalamus means everything under the thalamus, okay? And we have, of course, nuclei, okay? But other structures that shouldn't be forgotten, that they belong to the hypothalamus. And here we have the optic chiasm. As you see, the optic chiasm, this is a ventral view, okay? Optic nerves, optic chiasm. The optic tract forms part of the hypothalamus as well. The mammillary bodies, right here, remember they receive the columns of the fornix. We have the tuber cinereum. This is a gray matter elevation, right? And the infundibulum, okay? Infundibulum means a fune, okay? It's a part of the neurohypophysis. So in this image is an MRI in a sagittal section. You see here the optic chiasm, the hypophysis, right? So everything here is hypothalamus, okay? The jugum sphenoidale here, and then you have the optic nerves and the optic chiasm. And in this image, again, you see the optic nerve, hypothalamic nuclei, and inside the sella turcica, the hypophysis, okay? What it means, hypophysis? Hypo is under, physis is to grow. So anything that grows under. Okay, Andreas Vesalius thought it was the site to produce mucosity. This is why it is also named pituitary gland, from pituita. Then you have a ventral view of the encephalon with some of the cranial nerves, the eyes, the optic nerves, the optic chiasm, and here behind the optic chiasm, there we have the hypophysis, surrounded by cranial nerves. We will see that. Okay, so here we have the tuber cinereum, part of the hypophysis, part of the hypothalamus, the optic chiasm, part of the hypothalamus, the beginning of the optic tract, part of the hypothalamus, and the hypophysis. So this, this tuber cinereum is an elevated area that gives rise to the neurohypophysis, one of the parts of the hypophysis, the neurohypophysis. This neurohypophysis is formed by an infundibulum, a fune-shaped structure, and a neural lobe, right? So on a transverse section on MRI, you see the basal sphenoid here, and this hyperintense signal corresponds to the hypophysis. This is a mid-plane section where you see the interthalamic adhesion right here, the mesencephalic aqueduct, the third ventricle right here, the columns of the fornix, the rostral commissure, the optic chiasm, and the mammillary bodies. This is one of the mammillary bodies. There are two, one on each side is a pair structure. And then we have the hypophysis here with this funnel-shaped structure named infundibulum. So now, in a video, you have a better view of the hypophysis. This is the hypophysis right here. These are vessels of the encephalon we'll talk about in a next lesson. Okay. This is the hypophysis. Through this sulcus enters the diaphragma celli, okay, is a structure of dura mater, okay, diaphragma celli. So this is the aspect of the hypophysis in a dissection. It's very difficult to remove the encephalon with the hypophysis because it used to be attached. Okay, this is the image of the floor of the cranial cavity. The optic nerves right here, the internal carotid arteries, some cranial nerves. This one in particular is the oculomotor, right? So we see here, this is the sella turcica, the dorsum celli, and you see inside the sella turcica, the hypophysis. However, as you identify, there are different colors. This part, more brownish, corresponds to the neurohypophysis, that is the smaller portion. And this one that is more clear is the adenohypophysis. Once the hypophysis is removed, we see this dotted line marks the diaphragma celli. That is a part of the inner layer of the dura mater. So on a transverse section again, the interthalamic adhesion, hypothalamus, right? And here you have two portions, the neurohypophysis and on top the adenohypophysis. And if we make a midplane section, we identify the mammillary bodies, caudally located, always caudally located to the hypophysis. And then this is the tuber cyanurium, this gray matter elevation that gives rise to the infundibulum, right? Also named median eminence. This is important to know the names because sometimes in the papers there appear these names. So remember, this median eminence is the base, the most proximal portion of the infundibulum. And then continues and forms the neural lobe, right? The neurohypophysis. And on top sits the adenohypophysis. Sometimes we may find a cavity. It's the cavum hypophysis. This is the remnant of the Radke's bursa that originated. So you know that the adenohypophysis originated from the roof of the oral cavity that forms the Radke's bursa. So detaches and attaches to the neurohypophysis. Okay, this is the adenohypophysis. So while we are removing an encephalon from the cranial cavity, we identify the optic nerves, right? The hypophysis. Which part of the hypophysis is this one? The adenohypophysis, because you know the adenohypophysis is the most ventral portion, right? And the pons, etc. And then you have here the dorsum celli and the sella turcica and the dorsum celli. And this elevation of dura mater is the diaphragma celli. Look how close is the hypophysis to the optic chiasm and to the internal carotid arteries. Again, here we are going to, this encephalon was removed with part of the dura. So we remove this part of the dura that forms the diaphragma celli. And then we found the adenohypophysis. And then as we move the adenohypophysis with the forceps, we identify this is the tuber cyanurium, right? And then we have also, it is known as the pituitary stalk, right? This pituitary stalk, or also named infundibulum, remember that is this, is a part, okay, is a funnel-shaped structure that enters the hypophysis and it is surrounded by the neurohypophysis, okay? All right. Right. This is a video where you see, this is the floor of the cranial cavity, right? With the optic nerves, the internal carotid arteries, cranial nerves, and this is the inner layer of the dura mater. And as you see, there are different colors. You have this color here that is in the middle, that seems to be more green. It is the adenohypophysis with this hole is part of the infundibulum, right? And then on the outside, this more brown color is the adenohypophysis. And we are going to remove it. And as you see, the diaphragma celli is quite soft, even though this cranium was, or this head was fixed before dissection. Sometimes there are adherences, right? And it's difficult to remove it in a dissection. It used to break or detaches from the rest of the brainstem. Okay, there you have it. We are removing it. Okay, we are going to run fast, and you see the adherences here. All right, so we have removed the hypophysis from the sella turcica. Again, important, this region is very, very important, okay? Again, what do we have here? We have the diaphragma celli, this extension of inner layer of the dura mater, right? And the internal carotid artery, the oculomotor nerve, the trochlear nerve, the abduction, and also we should have marked here the trigeminal nerve. Okay. So an important thing to remember is that the hypophysis is not surrounded by the leptomeninges. Very, very important. It is just surrounded by dura mater, not leptomeninges. And as you can see here, this is an enlarged area. You see all the vessels running in the subarachnoid space, the internal carotid artery that has to go through these leptomeninges in order to enter the brain, right? Because this internal carotid artery comes from outside, okay, and has to enter this subarachnoid space, has to go down, right? Remember that the hypophysis is not surrounded by leptomeninges. And here again, we are going to do is to remove the hypophysis, and as we cut it through the infundibulum, we see the third ventricle inside. And then, as you can see, we can move the arachnoid from the pia mater, and this is the subarachnoid space where all the vessels are located. So, going about this diaphragma celli, what is that? Remember, here in this drawing, I have marked you the hypophysis and the rest of the hypothalamus on top. On both sides, we have the cavernous sinus and many other structures. So this is the paracella region with all the cranial nerves invaginating this dura mater inside or very in relation with the cavernous sinus, also vessels, the internal carotid artery in this case. Okay, this represents a medial plane section, okay? And as you see, in red is the pia mater, in green is the arachnoid, and as you see, the leptomeninges do not cover the hypophysis. Even though part of this internal layer, this pia mater, also runs on top of the neurohypophysis. Why so? Because the neurohypophysis develops from the neural tube, not the adenohypophysis that is coming from the Radke's bursa, right? And in blue, we have the dura mater. As you see, the dura mater, you know, is divided into layers, an inner and an outer layer. The outer layer is the periosteal layer. And then here, the inner and the outer layers split, right? And the inner layer forms the diaphragma celli. And the outer layer remains attached to the bone. And sometimes there are adherences. This is why sometimes it's difficult to remove the hypophysis without breaking it through the pituitary stalk. And in this space between these two layers of dura mater, there are veins that are going to receive the hormones. All right, again, we go to the dissection room and we make a mid-plane section, right? You see the presphenoid, basiosphenoid bone, basio-occipital bone, right? You identify the cerebellum, the cerebrum, and the brainstem here. We will focus here in this area. We see the, this is the dorsum celli, right? Remember that this head has been dehydrated, so the spaces are much bigger, right? And as you see, the dura mater and the subarachnoid space, and this space between the two layers of dura is bigger than it should be, right here. So remember, okay? And then you see this, the hypophysis located here, and very close to the, here we have the mammillary bodies and the optic chiasm, right? Okay. So, again, you know that you identify usually the hypophysis on MRIs. This is a T1 enhanced and T2 enhanced sequences. So, here we are, and when we want to identify the hypophysis, we have to cut at the level of the round foramen. Here is the alar canal, right? Here is the alar canal. This is an enlarged image here, and T1 and T2. And you see this is hyperintense signal corresponds to the hypophysis. This is the basi-sphenoid bone. In T2, we do not see hyperintense signal. And also, what we identify is the CSF in the subarachnoid space. This is the CSF in the subarachnoid space, because the subarachnoid space is located dorsally, right? Not, you remember, the hypophysis is out from the subarachnoid space. It's not inside. It's not covered by the leptomeninges. So it means that there is not CSF around the hypophysis, okay? So the thing is, sometimes, why it is bright? Why is hyperintense? It depends. It could be the anterior or a pituitary lobe, or adenohypophysis, or the posterior, depending. So in the case of T1, it could be the hyperintensity of intracellular proteins, concentration of proteins. Sometimes in a liver disease is the presence of circulating manganese. Or in the case of the neurohypophysis, the storage of vasopressin in the posterior or in the pituitary lobe or neurohypophysis. This is due because this hormone bounds to a macroprotease structure, and this is why this macroprotease structure shortens T1 signal and it's bright. So when you see hyperintense, could be the adenohypophysis or the neurohypophysis. In the case of a cat, it's quite similar. Okay, you see then is the dorsal section, right? These are the piriform lobes, and then you see the hypophysis. Then you see the hypophysis located here. But remember, the hypophysis is part of the hypothalamus. The mesencephalon. Mesencephalon is divided into portions, the tectum and the cerebral peduncles. Here you have a transverse section of a dog's brain at the level of the mesencephalon. Okay, what do you have? First, before going to the mesencephalon, what do you identify here? This is the corpus callosum, the splenium of the corpus callosum, right? The cingulate gyrus, cingulum, the hippocampus right here, okay? And then you see here the mesencephalon. Mesencephalon is divided in a tectum, here in red, that is formed by the lamina quadrigemina, that is formed by the two rostral colliculi and two caudal colliculi. And we have the cerebral peduncles. This is a pair structure. We have one on each side. And ventrally, the cerebral peduncles form the crus cerebri. Here, in this image, is a ventral image of a dog's brain, and I have marked in yellow the part of the mesencephalon, and this part is the crus cerebri because it's the ventral portion of the cerebral peduncles, right? We will start by the tectum and pretectal area. The tectum is formed by the two rostral colliculi and two caudal colliculi, okay? The rostral colliculi are involved in visual reflexes, and the caudal colliculi are involved in auditory reflexes. And here, just rostral to the rostral colliculi, we have the pretectal area, just caudal to the caudal commissure. Remember that the caudal commissure belongs to the epithalamus, that is the encephalon. However, the pretectal area belongs to the mesencephalon, to the tectum mesencephalic. Okay? The colliculi, they have a commissure between them, between the two rostral and between the two caudal. Okay, it's very easy to identify externally the commissure between the two caudal colliculi. Right here, the commissure. This is the commissura of the caudal colliculi. All right, and under the tectum we have the mesencephalic aqueduct. This is the mesencephalic aqueduct with the periaqueductal gray matter. Again, on top you see the commissura of the rostral colliculis. However, this one is smaller than the commissura of the caudal colliculi. You know that the pretectal area is important, right, in visual pathways, right here. Okay. There you have another image. This one here is that I mark is the pretectal area. Okay. In a sheep, this is in the case of a sheep, and you see that the rostral colliculus is quite big. Here you have the caudal colliculus. If you compare it with dogs, the rostral in this case seems to be much bigger. And there you see the pineal gland, sorry. This is the pineal gland. So in a transverse section, the caudal colliculus and then a big rostral colliculus. Okay. Okay, we are still in the mesencephalon. It is around the mesencephalic aqueduct, we have the periaqueductal gray matter. In further lessons, we'll see the importance of the periaqueductal gray matter. Then, this portion of the ventral portion of the periaqueductal gray matter is going to form the dorsal longitudinal fasciculus. This fasciculus is going to extend ventral to the fourth ventricle and down and caudal up to the spinal cord in the cervical segments at the level of the dorsal gray commissure. Here, this one here. So again, in the mulligan, you see the periaqueductal gray matter, right? And in this section, it is not a perfect one because as we see in the dorsal portion, we see the mesothalamus with the lateral geniculate bodies, the medial geniculate body, but ventrally we see mesencephalon. And which structures we identify in the mesencephalon? This one here. That is the red nucleus. Red nucleus. Remember this nuclei because in another lesson, we are going to talk about sensory motor systems. So these nuclei are going to be involved. Then we have the substantia nigra here, and we have talked about it because inhibition cells or inhibitory cells, GABAergic cells from the substantia nigra are going to be projected to the brainstem nuclei to control saccadic movements, and dopaminergic neurons are going to be projected to the basal nuclei to control its action. So lesions affecting the substantia nigra may disrupt or may damage the correct movements, right? So again, here you have the substantia nigra, and ventral to the substantia nigra, we have the crus cerebri formed by corticopontine and corticospinal fibers. And in between, in this level here, we have the interpeduncular nucleus. Remember that this nucleus was connected with the avenula. Again, in this image, you see the periaqueductal gray matter around the sylvian or mesencephalic aqueduct, and the ventral portion is the dorsal longitudinal fasciculus. This image is a very nice image, right? Because here we are at the level of intercrural fossa, right here. So we have at this level, this is the crus cerebri, right? Crus cerebri here with the substantia nigra. And here there is a nerve, is the oculomotor nerve. We will see that the oculomotor nerve, the nucleus, is located at this level, right? Ventral and very close to the midline, ventral to the periaqueductal gray matter. Okay, here. And their actions live in the intercrural fossa. They live, these actions live, and the oculomotor nerve. Remember that the oculomotor nerve has a somatic and visceral components. Okay, again in the mesencephalon. First, in this image here, you see that lateral to the periaqueductal gray matter, we have a nucleus. This is the nucleus of the mesencephalic tract of the trigeminal nerve. We'll talk about it, this nucleus, later on in particular. Nucleus of the mesencephalic tract of the trigeminal nerve, right? Is for proprioception. Okay, from the mandibular nerve. Again, this nucleus is located here. You see these cells right here. Again, you see the cells correspond to the oculomotor nucleus, right? Nucleus of the third cranial nerve. And ventral, the medial longitudinal fasciculus. This fasciculus connects vestibular nuclei with nuclei that move the eye, the extraocular muscles, right? In the movements of these are studied in the movements of the nystagmus. And there's also the medial longitudinal fasciculus continues in the spinal cord at the cervical segments in the ventral funiculus. So it's connecting vestibular nuclei with nuclei of the oculomotor system, right? With the oculomotor, trochlear, abduction, right? And also is going down in the spinal cord in the ventral funiculus. Then we have here the tegmental decussation, right? The tegmental decussation. This is a decussation of fibers. I have talked, or I have said before, that there was a tract, the tectospinal tract. We'll see this tract later in another lesson. The tectospinal tract starts at the level of the rostral colliculus. It is joined by fibers from the caudal colliculus, and their fibers cross the midline at this point, at the decussation of the tegmental decussation, right here. And they go down, right? Forming the tectospinal tract, okay? And ventral, at this ventral portion of the tegmental decussation, we have another decussation. Fibers coming from here. This is the red nucleus. It's a nucleus that belongs to the extrapyramidal system. Okay, it's a motor nucleus. The red nucleus is this one here, is the red nucleus. Their axons cross the midline at the ventral portion of the tegmental decussation, and they go down into the spinal cord in the lateral funiculus, forming the rubrospinal tract. Okay. And on top of the substantia nigra, we found the spinothalamic tract. This comes from the spinal segments and reaches the thalamus for nociception. And medially to it, the medial lemniscus and the trigeminal lemniscus. Right? Here, they also send information to the thalamus. We'll see these tracts later on applied functionally. Okay. So again, as we move, this section was done at this level. Okay, it's not perpendicular, right? So what do we see? On top, we see the rostral colliculi, right? Rostral colliculi of the mesencephalon. We see the mesencephalic aqueduct and the periaqueductal gray matter. And ventrally, we are cutting the pons, right? The pons. This is a cerebellar peduncle and the trigeminal nerve right here, okay? This is the trigeminal nerve. It should be going rostrally, right? So at this level, because we are a bit more caudal than the first section, we found the trochlear nucleus, the motor nucleus of the trochlear nerve, right? Okay. And as well, as we go into histology, periaqueductal gray matter. Here we have the dorsal longitudinal fasciculus. Remember the nucleus and mesencephalic tract of the trigeminal nerve, the nucleus of the mesencephalic tract of the trigeminal nerve, right? And you see here, this one here is this structure, is the medial longitudinal fasciculus, ¿vale? Medial longitudinal fasciculus. Remember that this fasciculus is connecting nuclei that move the eye with the vestibular nuclei, right? And right here, this is another decussation. This is the decussation of the rostral cerebellar peduncles. As we will see when we will talk about the cerebellum is connected to the brainstem through the rostral, medial, and caudal cerebellar peduncles. The rostral one is formed by fibers that leave, the fibers that leave through the rostral cerebellar peduncle, right? They cross the midline at the decussation of the rostral cerebellar peduncles, and they go to the prosencephalon, okay? We reach the level of the pons, right here, the pons. And again, a transverse section. So we are cutting at the level at here, at the level of the caudal colliculi, right here. In the middle, we have part of the cerebellum. And then eventually we have the pons with the middle cerebellar peduncle that connects the pons to the cerebellum. And laterally, we have the trigeminal nerve. Okay, right. So this is a Bialchowski, right? And what do we see here? Well, eventually, the pons is located here, caudal to the mesencephalon and rostral to the medulla oblongata. So again, the caudal colliculi, in the middle, the vermis of the cerebellum. And here, again, this area is the continuation of this one that was located ventral to the mesencephalic aqueduct, that is the dorsal longitudinal fasciculus, also named Schuth fascicle, okay? That runs between hypothalamic nuclei and general visceral efferent nuclei of the brainstem, okay? So it's very close to this, related with the hypothalamus. And ventral to it, the medial longitudinal fasciculus that we have talked previously. On top, there is a very important nucleus, okay, because it's one of the micturition centers. It's the locus caeruleus, okay? It's a collection of adrenergic neurons, okay, that store, synthesize, and release noradrenaline, okay? It is also known as the pontine micturition center, that related in the voiding the urinary bladder, okay, in the detrusor reflex. Here, the mesencephalic tract of the trigeminal nerve. Remember that it's gone up and it's going to reach this nucleus. It's going to reach this nucleus here, the nucleus of the mesencephalic tract of the trigeminal nerve, located lateral to the periaqueductal gray matter in the mesencephalon. Other structures that are very interesting is the rostral cerebellar peduncle. Remember that the cerebellum is connected through the rostral cerebellar peduncle with the mesencephalon, and the fibers that leave cross the midline in the, here, as I have said, in the decussation of the rostral cerebellar peduncles. Another important, the rubrospinal tract. Remember that these fibers have crossed the midline at the tegmental decussation in the mesencephalon, and they are coming down in order to reach the spinal cord. Is the rubrospinal tract, okay, in the lateral funiculus. What else? The lateral lemniscus. This lateral lemniscus is a tract of fibers that is coming from the dorsal nuclei of the trapezoid body for audition. They are auditory, acoustic fibers, and they are going up and they reach the caudal colliculus. And remember, when we were talking about the metathalamus, the caudal colliculus receives acoustic information, and this acoustic information is projected, okay, to the medial geniculate body, okay, of the metathalamus, and from there it goes to the temporal cortex. Again, the spinothalamic tract for nociception and the medial lemniscus that sends information here, this structure, this sends information for proprioceptive information to the thalamus. Okay, this is a mid-plane section on MRI in a T2 sequence, and it's important we are going to focus in this area. Where are we? Mesencephalon, right? This is the tectum, the mesencephalic aqueduct. Remember that the mesencephalic aqueduct does not belong to the tectum. It belongs to the crus cerebri, to the brachium of the mesencephalon. As you see, remember, go back here, right, to the cerebral peduncles. Look at this. The mesencephalic aqueduct belongs to the cerebral peduncles, not to the tectum. Okay, so, and then the pons and the medulla oblongata, right? See how the mesencephalic aqueduct opens caudally. However, it has the cerebellum, but is separated by the roof of, at this point, the roof of the fourth ventricle, it is very thin. And so the cerebellum, it is not in contact with the CSF inside this mesencephalic aqueduct and fourth ventricle. There is a thin lamina. This one here, there is a thin lamina, right? Is the roof of the fourth ventricle, right? Okay. Right. It is also named the rostral medullary velum. Okay. Okay, this rostral medullary velum is like that. Okay, there is the caudal colliculi, the commissure of the caudal colliculi here. So we have the opening of the mesencephalic aqueduct, and then this is part of the cerebellum. So this is to show you the rostral medullary velum that is closing the mesencephalic aqueduct caudally. And remember, which structure we found here? We have the decussation of the trochlear nerve at the rostral medullary velum, just here, just caudal to the opening of the mesencephalic aqueduct. Okay. So we still are in the pons, okay? This is a sagittal section where you see and transverse sections. What I want you to point is that the pons, the ventral portion of the pons right here, because we have talked about that, remember, right? The spinothalamic tract, the medial lemniscus, the lateral lemniscus here, right? And ventral to it, we have the ventral portion that is formed by longitudinal fibers. Look at these fibers. These are longitudinal fibers. They are cortico-spinal fibers, corticopontine fibers, right? These are the longitudinal fibers. And among them, there are pontine nuclei, okay? This large area is here with a Balchowski. And these pontine nuclei receive information from prosencephalic structures and send this information through the middle cerebellar peduncle inside the cerebellum. Okay? Okay. So we are still in the pons, but in the caudal portion of the pons. And now I want you to show you something is important. Two nuclei. Number one here is the sensory nucleus of the trigeminal nerve, located rostral to the spinal tract of the trigeminal nerve. This receives pain and temperature. And the other one, medially located, is the motor nucleus of the trigeminal nerve, also named masticatory nucleus. Okay? Important. And in histology, you have it here, 1 and 2. Okay? The sensory nucleus of the trigeminal nerve and the motor nucleus of the trigeminal nerve, or masticatory nucleus. So again, we are at the level of the pons, remember that. Okay, look at these connections with the cerebellum, the middle cerebellar peduncle, the rostral. We'll see that when we'll talk about the cerebellum, right? And again, what you have here, this is the fourth ventricle. Okay, this is the floor of the fourth ventricle, the cerebellum there. And remember, this is the dorsal longitudinal fasciculus. It's a fasciculus that comes from the periaqueductal gray matter that has opened, right? And continues caudally and connects a hypothalamic nuclei with general visceral nuclei. Okay, right. Again, this is the dorsal longitudinal fasciculus here. We are at the level of the pons. Remember, this is the ventral portion, right, with the longitudinal fibers, transverse fibers that connect the pontine nuclei with the cerebellum through the middle cerebellar peduncle here, right here. Okay, and the dorsal portion. Remember, the medial lemniscus, spinothalamic tract, lateral lemniscus here. We reach the medulla oblongata. That means more long than wide. Okay, and again, dorsal, ventral, and raphe. Here I have marked in yellow the medulla oblongata. So this is the pons, the mesencephalon, this is the crus cerebri, right? The ventral portion of the cerebral peduncles, right? Okay, trigeminal nerve here, right? The abducens nerve here, and these are the pyramids, the trapezoid body, and the rest of the medulla oblongata, up to here, up to near the decussation of the pyramids here, right? So this is a ventrolateral view where you see the trapezoid body related with the facial vestibulocochlear nerves. Here, the glossopharyngeus and vagus, right? The accessory, right? Here, okay? And here you have the hypoglossal nerve. Okay, and this is the medulla oblongata with eventually the pyramids. So we'll start by the trapezoid body. That means like a trapeza, okay? From the Greek, a table. It is this portion here that is ventrally covered by the part of the pyramids. Remember that the pyramids are formed by the cortico-spinal fibers right here. Okay, transverse section again, the pyramids right here, and these white fibers here, this is the facial and vestibulocochlear nerves, and these white fibers here form the trapezoid body. These are these transverse fibers that are easily seen on a dissection on both sides of the more rostral portion of the pyramids. So, okay. And what do we have at this level? Here, it's in this transverse section, you see the lingula, part of the vermis, the cerebellar hemispheres, and then facial and vestibulocochlear nerves, right? And these white fibers, the trapezoid body, top of the pyramids, and continue right here, okay? And there, this structure is the dorsal nucleus of the trapezoid body, formerly known superior olivary nucleus. Remember this name because it's written in some books, classic books, right? So the dorsal nucleus of the trapezoid body. And when I mean dorsal nucleus of the trapezoid body, there is a ventral nucleus. And the ventral nuclei are scattered among the trapezoid body. Trapezoid body are the transverse fibers right here. Okay, this transverse fiber here. So this is the dorsal nucleus of the trapezoid body. We will see the function now. And now we start to see the spinal tract of the trigeminal nerve and the nucleus of the spinal tract of the trigeminal nerve here. Remember that we are at the fourth ventricle here, and what do we have? We have at this level, we have vestibular nuclei, right? The medial, right? Vestibular nucleus, the lateral vestibular nucleus, and this is the caudal cerebellar peduncle. This big is the caudal cerebellar peduncle. Okay, and the pyramids. And again, important, this is the motor nucleus of the abduction. Remember that the abduction cranial nerve, where was it? Here. The abduction cranial nerve was coming on this level, you see, between the pyramids and the trapezoid body, and runs rostrally. Okay, so there you have the motor nucleus of the abduction, right? And here the genu of the facial nerve, because the facial nerve goes around the motor nucleus of the abduction cranial nerve and forms the internal genu of the facial nerve, and then leaves here between the rubrospinal tract and the spinal tract of the trigeminal nerve, and leaves. The facial fibers leave here. Okay, so again, this is a complete image, right? You have here, remember the dorsal nuclei or nucleus of the trapezoid body, formerly known as superior olivary nucleus. And again, the rubrospinal tract, look where it is a motor tract, right? And this is a sensory spinal tract of the trigeminal nerve with the nucleus of the spinal tract of the trigeminal nerve, right? And here as well, the medial longitudinal fasciculus, because it's connecting vestibular nuclei with nuclei for the movement of the eyes. So let's go to this tract that is very, very important. This is a summary, okay? And what it represents. Here you see very easily on a dissection, right? You see the spinal tract and the nucleus. And this spinal tract is a somatotopically organized. So somehow the fibers that go to the most dorsal portion are from the mandibular, on the dorsolateral from the maxillary nerve, and ventral from the ophthalmic nerve, right? And here we have the nucleus of this spinal tract of the trigeminal nerve. And again, okay. So what I have done here. So this, remember that in the previous images, I have shown you the masticatory nucleus, right? The motor nucleus of the trigeminal nerve, and with a relation with the sensory nucleus of the trigeminal nerve, included in the most caudal portion of the pons. And this was receiving, through the semilunar ganglion, information from pain, or nociception, and temperature. And now here you have it, the ones that are in the spinal tract, the ones that are receiving tactile and pressure, right? But also from all the cranial nerves, general somatic afferent, from the facial, from the glossopharyngeal, from the vagus, they reach here. So this spinal tract is a kind of pathway to distribute information in order to enter the nucleus of the spinal tract of the trigeminal nerve, and to be able to respond to other reflexes and functions, right? And it's important to notice that this spinal tract of the trigeminal nerve continues in the spinal cord, and it does so as the dorsolateral tract. And the nucleus of the spinal tract of the trigeminal nerve continues in the spinal cord as the substantia gelatinosa. So it's a continuation between brainstem and spinal cord, right? It's important to notice that. Okay. So we continue, and then what do we see? We see that we are at this level now. Okay. This is the dorsal cochlear nucleus. Comes from the cochlea on the inner ear. And these are these superficial arcuate fibers that go on top, these fibers here, that go on top of the caudal cerebellar peduncle. And we will see where they go, okay? to the dorsal nuclei of the trapezoid body and to the audition, etc., etc. Okay, and oh, sorry, here you have it. So this is the caudal cerebellar peduncle, and on top you have this. These are the acoustic stria. The acoustic stria here is formed by fibers that are coming from the dorsal cochlear nucleus, and they go on top of the caudal cerebellar peduncle. Okay. So again, now we are having a look at the brainstem, and on top of the cerebellum, I have to move part of the flocculus in order to identify the acoustic stria. You see, this is the vestibulocochlear nerve, right? And so the acoustic fibers are going on top of the caudal cerebellar peduncle here. They form the acoustic stria. The acoustic stria jumping on top of the caudal cerebellar peduncle, right here. And where do they go? Right. This is a scheme that I think it will help you understand. Okay. So fibers from the dorsal cochlear nucleus, they go inside, they form the acoustic stria right here. This is important area, and then they go down, and they reach the dorsal nucleus of the trapezoid body. Okay, and from there, some fibers they go up, or ipsilateral or contralaterally in the lateral lemniscus in order to reach the caudal colliculus. Other fibers from the ventral cochlear nucleus form the trapezoid body, and the ventral nuclei of the trapezoid body are scattered among the trapezoid body. These fibers, they enter the ipsilateral and contralateral nuclei, the dorsal nuclei of the trapezoid body. And again, these nuclei project through the lateral lemniscus to the caudal colliculus. Some fibers from the dorsal nucleus of the trapezoid body, what they do is they do the opposite. They run with the acoustic stria and they go to the ear. This is the olivocochlear fascicle. And this is to modulate the cochlea, right? And this is important because the vestibulocochlear nerve, we always think that it's a sensory nerve, but it's not true because it has motor fibers forming the olivocochlear fascicle. And remember, this olivo because the dorsal nucleus of the trapezoid body was formerly known as rostral olivary nucleus. Okay, this image, I have added this one because it's not a true one. Here, yes, we have the acoustic stria on top. However, at this level, these fibers, they go rostral and they reach here, we have the dorsal nucleus of the trapezoid body. The dorsal nucleus of the trapezoid body is more rostrally located than the acoustic stria. Okay, so if we move caudal in the medulla oblongata, here you have the fourth ventricle, the cerebellum right here, okay, the spinal tract of the trigeminal nerve, rubrospinal tract, we know it is there, the caudal cerebellar peduncle, acoustic stria right here. So we find that this is the motor nucleus of the facial nerve, very close to the rubrospinal tract. The rubrospinal tract is here located, ventral to the spinal tract of the trigeminal nerve. So these fibers from the motor nucleus of the facial nerve, they go up, they go around the nucleus of the abducens nerve. They form what is known the internal genu of the facial nerve, and they go down to live between the spinal tract of the trigeminal nerve and the rubrospinal tract. Okay, this is the motor nucleus of the facial. And remember again, we have the medial lemniscus and the spinothalamic tract, but at this level, we do not find the lateral lemniscus because the lateral lemniscus has started in the dorsal nucleus of the trapezoid body that is located rostral to this section. Okay, we proceed in the medulla oblongata. This is the medulla oblongata on MRI. This is the basio-occipital bone, right? The fourth ventricle here, the pyramids, right? And this is in a dissection where you see the spinal tract of the trigeminal nerve, right? On top, we have the caudal cerebellar peduncle, right here. You see the caudal cerebellar peduncle and the spinal tract of the trigeminal nerve. In Abialchowski, again, you see the caudal cerebellar peduncle, the spinal tract, and here you have part of the stria acoustica still, right? And you see the fourth ventricle filled with the choroidal plexuses of the fourth ventricle. Some of them are living through the lateral openings of the fourth ventricle. We proceed more caudally here at this level, and at this level we begin to identify the olivary nucleus. Here you have the olivary nucleus, lateral to the pyramids, the olivary nucleus. Okay, and some fibers coming out from the olivary nucleus, they form the olivocerebellar tract, fibers that cross the midline and enter the cerebellum, right? They belong to the extrapyramidal system. Okay, again, the deep arcuate fibers, they form the decussation of the medial lemniscus, right? The medial lemniscus, we will see later on, these fibers are coming from the nuclei as the cunate nucleus, gracile nucleus, Z nucleus, etc. So at this level, we found the motor nucleus of the hypoglossal nerve. And these fibers are coming down. Look, the nucleus is very close to the floor of the, or is in the floor of the fourth ventricle, right? Very close to the ventricle. And the fibers are coming down, and living between the pyramids and the olivary nucleus. You see, between the pyramids and the olivary nucleus, there are the fibers that are coming out and form the hypoglossal nerve. Also, we found other nerves, the solitary tract and the nucleus of the solitary tract, right? We will see that in visceral functions, right? When we'll talk about the cranial nerves, remember, the solitary tract and the nucleus, right? And also the caudal cerebellar peduncle, and also still some of vestibular nuclei here, okay? Because we are, but very, very extension, very caudal extension, right? So again, the spinothalamic tract located ventrally. So it's quite superficial, this spinothalamic tract. This is a general view of the medulla oblongata, right? With all the structures. Remember for the pathologists may need the solitary tract, the nucleus of the solitary tract, where it's located, right? Okay. And as you see, all these the commissural fibers, right? The olivocerebellar tract and the decussation of the medial lemniscus, okay? Forming the deep arcuate fibers, here, okay? Deep arcuate fibers. So again, we go a bit more caudal. We are at the level of the obex, here, okay? The fourth ventricle, right? And then the fourth ventricle becomes a canal, right, that will continue as the central canal of the spinal cord. So we are at the level of the area postrema here, area postrema. And important to notice what you have here. You have the gracial fascicle here, right here, on top. And here, the spinal tract of the trigeminal nerve. The spinal tract of the trigeminal nerve. So this spinal tract of the trigeminal nerve can be easily identified from the outside, right? Look here. Sorry, this is the spinal tract of the trigeminal nerve. This one here is located here, sorry. And this is the gracile and the cunate, right, and fascicles here. Okay. So important, you see the motor nucleus of the hypoglossal nerve right here, right? Important other structures, the nucleus ambiguous, right? And the ventral reticular nucleus located. These cells belong to the ventral reticular nucleus. And again, look at this position. Lateral to the motor nucleus of the hypoglossal is the parasympathetic nucleus of the vagus nerve. So at this level again, we see the gracile fascicle, the cunate fascicle here, and the spinal tract of the trigeminal nerve and spinocerebellar tracts. So externally, it's easy to identify them, very, very easy, right? And also all these fibers that jump and cover the spinal tract of the trigeminal nerve, these fibers here, are the superficial arcuate fibers. These are fibers that are leaving the lateral reticular nucleus, and they join the caudal cerebellar peduncle. All right, here. So in a transverse, you see the gracile fascicle. This is the gracile fascicle here. We see again the fasciculus cuneatus, or the cunate fascicle, and lateral. This structure is the spinal tract of the trigeminal nerve. Okay, with the nucleus of the spinal tract of the trigeminal nerve. And more ventrally, you have the spinocerebellar tract. Okay, so these structures can be identified externally. So again, you have this whole picture, right? And again, do not forget about the medial longitudinal fasciculus, and now the tectospinal tract located here, the pyramids and the olivary nucleus, and lateral to it, the spinothalamic tract. Okay? So now we are approaching a very specific area. It's the decussation of the pyramids, very important. But before, we are going to talk about this area on top of this section, right? What do we have? This is a Bialchowski, right? Impregnation, right? And the central canal, and on top, we see some collection of cells. Right? And they are, this one here is the Z nucleus, right? And the one that is more ventral and laterally located is the nucleus gracilis. So it has been considered the nucleus theta as part of the gracilis nucleus. It is involved in proprioception, right? In proprioception, particularly proprioception from the hind limb. And then these collections here are these ones here, and we are going to follow this to this that is marked in red. So we have the nucleus cuneatus medialis, or medial cunate nucleus, and the lateral cunate nucleus for proprioception, for conscious and unconscious proprioception. We'll see that in the pathways, when we'll study the pathways, right, of proprioception. Again, it's important to remember, this is again at this image, okay, and this image located around here, what do we see? We see structures that are important to remember. The motor nucleus of the nucleus, this is the nucleus ambiguous of the accessory, right? Also, we have the lateral reticular nucleus, okay? And the fibers from the lateral reticular nucleus, this area has been enlarged here. The lateral reticular nucleus projects fibers through the superficial arcuate fibers. They form the superficial arcuate fibers, right? and they are located on top of the caudal cerebellar peduncle, right? These are involved in posture, grasping, locomotion, scratching, respiration. Yeah, this is an important center. The lateral reticular nucleus and the ventral reticular nucleus here. Okay, so remember when we look this image, we are still seeing what? The spinal tract of the trigeminal nerve and the nucleus of the spinal tract of the trigeminal nerve, the nucleus and the spinal tract, right? And remember that these structures are continuous in the spinal cord as the Lissauer's bundle also, or the dorsolateral tract, this one here, and the substantia gelatinosa. So these structures are continued in the spinal cord, right? Okay. So important, again, all these fibers, we are at the level of the decussation of the pyramids, right? So remember that the pyramids were ventrally located. So now the fibers are going to go up, cross the midline, and then deviate laterally, as these arrow marks. This is the medial longitudinal fasciculus and tectospinal tract. Okay, they are going to form funiculi in the spinal cord, right? And these are fibers because it's a Bialchowski. They go up, and they locate here. These areas here belong to the beginning of this corticospinal tract in the spinal tract. Okay, these are fibers from the corticospinal tract that are beginning to be located in the location that they will have in the spinal cord, and here the rubrospinal tract. Okay, so again, in this image, you see that this rectangular area, it is enlarged here, and you see these are the fibers that cross the midline at the decussation of the pyramids. The pyramids are located there. The fibers go up, they deviate laterally, and here they begin to form these fibers that are going to go down in the spinal cord. Is the corticospinal tract, and ventrolaterally to it, we have the rubrospinal tract. Look at it. This again, in this area, you see these fibers, the pyramids, the fibers that go up, they decussate, form in the decussation of the pyramids, and they move laterally in order to form the corticospinal tract with its location in the spinal cord. And all these fibers here, that will become the ventral funiculus in the spinal cord. Thank you very much.